To mount a successful defense, the immune system must solve a basic spatial problem. Pathogens exist in two distinct physical environments, circulating freely in fluids like the bloodstream or hidden away inside a compromised host cell. A mechanism designed to intercept a virus in the blood cannot reach a virus replicating inside a lung cell. This physical barrier necessitates the adaptive immune system's split two-arm architecture. The first arm is humoral immunity. This is a fluid-based defense network mediated by B cells and their antibodies, engineered to target extracellular threats before they breach a cell membrane. The second arm is cellular immunity. Mediated by T cells, this system identifies and destroys intracellular threats, such as host cells that have become infected by viruses or turned malignant. Both arms operate simultaneously during an infection, but they deploy entirely different biological hardware for recognition and execution. Mapping the divide between the extracellular and the intracellular provides the foundational blueprint for modern clinical immunopharmacology. The first step in this dual architecture is the recognition phase. The humoral and cellular systems scan the body for threats using distinct biological rules. Humoral recognition utilizes B cell receptors as surface sentinels, binding directly to intact pathogens floating in extracellular space. The cellular system relies on MHC restriction. T cells ignore free-floating pathogens, only recognizing threats processed into peptide fragments and displayed on a host cell by an MHC molecule. The exogenous pathway handles external threats. Antigen-presenting cells engulf extracellular microbes, displaying fragments via MHC class II to activate CD4-positive helper T cells. Conversely, the endogenous pathway handles internal threats. Infected cells present viral proteins via MHC class I to alert CD8-positive cytotoxic T cells. This dual-class MHC system ensures the body deploys the correct type of force. It uses class II to coordinate against external threats, and class I to order the immediate destruction of an infected self-cell. Once a threat is recognized, the system enters the activation phase. But initial contact is insufficient to trigger a full immune assault. An immunological synapse requires three-signal verification. Signal 1. The key cell receptor binds the peptide MHC complex. Signal 2 is co-stimulation. A CD28 receptor binds a B7 molecule on the presenting cell. Without signal 2, the T cell enters energy, a non-responsive state preventing attacks on healthy tissue. Signal 3 involves cytokine release, dictating if the T cell differentiates into a Th1 or Th2 effector cell. The humoral arm operates under similarly strict verification. For B cells responding to protein antigens, binding the target is only the first step. In T-dependent B cell activation, the B cell internalizes the antigen and presents it to a CD4-positive helper T cell. To proceed, the B cell must receive a physical secondary signal via a CD40 to CD40 ligand bridge connecting the two cells. Once verified, the activated lymphocytes trigger massive clonal expansion, multiplying into an army of identical cells and undergoing affinity maturation to increase their binding precision. These multi-step checkpoints balance the requirement for exponential cell growth against the risk of an autoimmune response. With activation complete, the system moves to the execution phase, where expanded clones deploy their specialized weaponry. In the humoral arm, activated B cells become plasma cells. These biological factories secrete thousands of Y-shaped antibodies per second into the bloodstream and lymphatic fluid. Antibodies do not kill pathogens directly. Instead, they neutralize extracellular toxins by blocking their receptors, or they opsonize circulating microbes, coating them to enhance phagocytosis by macrophages. The structure of the heavy chain determines the specific physiological role of the antibody, from the pentameric IgM driving the primary response to the monomeric IgG handling widespread neutralization and secretory IgA protecting mucosal surfaces. On the cellular side, execution requires direct contact. To eliminate an internal threat, CD8-positive cytotoxic T cells firmly and physically engage their infected target cells. The T cell deploys the perforin-granzyme pathway, forming pores in the target membrane so granzymes can enter and activate a caspase cascade. Alternatively, the Fas-Fas ligand pathway uses receptor interaction to trigger the same cascade, resulting in programmed cell death, or apoptosis. These execution methods, fluid-based antibody neutralization and direct cell-to-cell apoptosis, are centrally coordinated by CD4-positive helper T cells releasing regulatory cytokines. The successful resolution of an infection generates long-lived memory cells. These cells ensure a rapid, high-magnitude secondary response if the same pathogen returns. This memory mechanism is the foundation for traditional vaccines and booster doses, which induce humoral neutralizing antibodies against viral and bacterial threats. In clinical application, we use highly engineered monoclonal antibodies, or MABs. These are manufactured effectors deployed to intercept and neutralize extracellular targets, commonly used in treating autoimmune diseases and specific cancers. Organ transplant rejection occurs when host T cells recognize foreign MHC molecules on donor tissue and attack. Immunosuppressants like cyclosporin intervene. By inhibiting the cytokine IL-2, the drug blocks T cell expansion, preventing tissue destruction. Next-generation cancer immunotherapies use checkpoint inhibitors to intercept the chemical suppression signals, like PD-1, that tumors use to hide. This hyperactivates cytotoxic T cells to resume their attack on the malignancy. CAR T therapy involves extracting a patient's T cells and reprogramming their receptors to hunt specific tumor antigens before infusing them back into the body. The adaptive defense functions as a highly specific, programmable, dual-arm network. Mastering the mechanics of humoral and cellular immunity provides the exact logic required to wield modern immunopharmacology.